To talk a little bit about animal models, something that's been bandied about throughout the sessions here, but now we'll focus on what can and can't be done with them. My pleasure to introduce Steve Perrin. Thanks, Steve.
Thank you, Ted, and thank you, Howard, for inviting me to present today. I must be honest, I was quite surprised by some of the topic this morning, and if I would have known about some of the folks' feelings on animal models, I actually do have a much more defensive postured position than this slide deck, but I'll try to get you through it anyway. And hopefully this is a Prezi, nobody gets too much vertigo. What I thought I would do actually to cover this topic is give you a historical perspective to try to put into place the challenges that we have with developing a validated animal model for a disease. And that's a really loose question right there, because a mouse is never going to be a person, period. But if you do want to try to increase your likelihood of pushing drugs forward, they have to have some aspects that replicate the human condition. And even though we think we've done a great job with that, I would question how well we've really rigorously done that. And I'll point out some examples here. So I'll start here. In 2008, ALS-TDI published a paper on how to design drug studies in a preclinical model of ALS, the SOD1 animal model. And the paper was quite controversial, and I'll throw out there the first question: why was it important? And I think to answer that question, we have to go back in time. So let's start off with how we got there. Well, way back in 1993, the first mutation associated with ALS was identified in the SOD1 gene. And it's a very small percentage of patients, about 2% of patients harbor this mutation. Interestingly enough, we still to this day don't know really what the cause of that gain of function is. We really don't have any understanding of it whatsoever, and it changes probably about every 4 to 5 years. But what was interesting is a few years later, people made the first transgenic, basically, and now there's about 8 or 9 different SOD1 transgenics that come in different flavors, different point mutations, different promoters. But when you overexpress the mutant human SOD1 in a mouse, they get progressive neurodegeneration that looks very, very similar to what we see in people. Typical hallmarks of dieback at the neuromuscular junctions, muscle atrophy, neural inflammation in the periphery as well as in the CNS, microglial activation, astrocytosis, protein misfolding, axon transport deficits. All of these things have been described in multiple animal models using SOD1 as a transgenic to drive neurodegeneration, and it's also been described in ALS patients. So here's the Kaplan-Meier survival plots from that first transgenic that was published about 4 years after the mutation was identified. And as you can see, it comes in a couple of different flavors. This is actually the same mutation, but depending upon how many copies of the mutant transgene you express, that increases the toxicity and changes the kinetics of the lifespan of the animal model. The more copies you express, the faster the animals die. And that was really key. If you fast forward back to today, this is our current animal model at ALS-TDI. We've been maintaining it since 1999. The kinetics are exactly the same, both for the high copy expressor as well as the low copy expressor. So the bottom line is, this transgenic animal model is very, very stable, and it's probably the most widely distributed animal model for ALS today. So what's the problem if it's that reproducible? Well, the problem is studies like this, where in a very well-respected journal from a very good lab, a drug was shown, and again, Kaplan-Meier plot, to extend survival in this model statistically significantly, increased lifespan by as much as 25%. A couple of things to keep in mind: 9 animals per group, and no other details about the study design. Were these male animals, female animals? Were they balanced between groups? Were genders, were siblings balanced between groups? When did dosing start? Very little information on the preclinical design, but because of this data here, fast forward a few years to a phase 3 clinical trial showing that Celebrex had absolutely no effect in phase 3 in ALS patients. Let's go back again. Minocycline. Again, paper shown, very high-tier journal, really great lab. 6 or 7 animals per group, statistically significant effect. Again, no information on study design. Genders, siblings, dosing, very, very little data. Fast forward, phase 3 clinical trial actually exacerbated disease in people, and they had to stop the trial early. And I'll go back once more. Lithium is the most recent example. Again, a significant extension in survival. Fast forward to the clinical trial, and again, the clinical trial was stopped early because it actually exacerbated disease in people. So that's really the problem. So let's go back to how we got here. Well, we published this paper because when looking at all of the literature for the last 10 years, studying drug effects in this model, it was apparent that nobody could replicate anybody else's work. And so these are the studies actually, because one of our missions as a nonprofit is every time somebody publishes a drug study in ALS, in the SOD1 model, we immediately call those authors and try to replicate it exactly like they described it. And here's our survival data in our hands for all 3 of those drugs. Celebrex didn't work at all. Minocycline actually exacerbated disease in our hands. It did not improve survival, much like in the clinical trial. And obviously lithium didn't do anything in our hands either. The accumulation of all of that is somewhat captured in the slide here, where in our hands, here's our % survival of about 25 drugs. We've now screened about 150 since I joined in 2005, of drugs that went to the clinic. We've wasted probably $800 million in clinical trials in ALS for drugs in our mind as an institute should have never gone there in the first place. And that's because the preclinical trials were done so poorly, the data was completely uninterpretable. And this really shouldn't be news. The Wall Street Journal wrote an article recently that probably as little about as 10% of preclinical work is actually validated by anybody else. And maybe the amount of rigor that we've heard today that we put into clinical trials needs to be pushed back into the labs that are actually doing preclinical work at universities, pharmaceutical companies, and even in nonprofits as a whole. I tell you, when I worked at either Sanofi or Biogen, if I had walked into our VP of neurology with N's of 10 for a drug and said we should push this drug to the clinic, they would probably fire you. So just something to keep in mind. So what did we do at that time? Well, we have the largest colony of SOD1 animals in the world, so we did historical analysis. This is basically every one of these dots is an animal in our colony over time. As you can see, the median survival of this colony is very reproducible. It's about 135 days. However, there's outliers, animals that live too long, animals that succumb to the disease supposedly sooner than most of the animals at median survival. And we went back and we looked at in our database, because when an animal comes in the door or is born at TDI, it gets barcoded and we track every single aspect of that animal all the way till death. When we went back and we looked at all of these animals, we started having the ability to figure out what are the outliers. So most animals that live too long have a variation in copy number. As I said earlier, copy number directly affects median survival times in this mouse model, and they do not like keeping 24 copies of that mutant human transgene when they have young and basically pass off that locus to their offspring. They tend to kick randomly 8 or 9 or 10 copies at a time, and therefore some of the siblings from that litter will live longer. And so if you don't regenotype every single animal before starting into a drug study, you start to have false positive animals that are living too long. We also noticed that in addition to copy number, a lot of animals that were dying too soon were not dying of ALS. They were dying of something that was happening in the lab, either too aggressive in a treatment when delivering drug, either IP or IT, an animal gets dropped on the floor, they fall off the rotor rod. All of those animals need to be excluded from an analysis, and although I hear chuckling, it's amazing that people don't censor their animals, as we do in clinical trials often. And finally, there are some gender differences. Male animals get sicker and have more aggressive disease than female animals, so if you're going to do study designs with drugs, you better balance your genders. And again, 90% of the publications don't even talk about what genders in their studies. So these are all variables that need to be controlled. And we basically published a Monte Carlo analysis of our data set to ask the question, if you don't control any of those 4 variables, how often would you detect a fake 5% drug effect? These are all animals that were never treated with drug ever. They were from all of our control groups over the last decade. And interestingly enough, with N's of 4, and again, I've seen at least 20-something publication with N's of 4 published in journals like Nature, 60% of the time you can detect a 5% drug effect if you don't control any of the variables, and if you can control all 4 of them, you'd still detect 30%. So you need to get down to N's of 20 to 30 to get your false positive rate down to a reasonable level of a couple of % in a study. And this was published again back in 2008. Here's the study design. And again, this starts to address and even look like what one might expect in an actual clinical trial. And if you're going to study drugs in an animal model, these are the types of variables that you need to look at and control. Now, when we put this paper out in 08, we actually have upped this recently because this allows you to detect a drug effect when you combine genders. So this is a combination of genders. If you want to detect a drug effect in separate genders, males and females, you actually need a little bit more power to do that. You have to go up to 16 animals per per group, 16 males and 16 females, so 64 mice total. And again, we only learned that because as we start to finally get some positive hits in drug screening in this model, there are gender effects. Some drugs seem to work better in one gender versus the other. So let's fast forward again to a few years ago. A second mutation, highly correlated with ALS, a couple of percent, just like we see for SOD1, mutations in the TDP43 gene, was published in 2006. Looks very, very similar to what we see in patients with protein aggregation. These aggregates are actually present in 98% of ALS patients, much more so than the actual genetic mutation itself. A few years later, 4 years later, 2009, if you overexpress that human mutant TDP43 gene in mice, they get progressive neurodegeneration, as was published in PNAS. And they showed in a couple of mice that there's loss of motor neurons, muscle atrophy, and many of the other hallmarks that one has seen in the SOD1 model in ALS patients. So you should be thinking deja vu at this point. This is the Kaplan-Meier plot from that paper, and yes, they have decreased lifespan. But one of the things that's interesting about this decreased lifespan is, oops, I'm sorry, I clicked too fast, is that if you look at the kinetics, or the variability, I should say, in survival, a simple power analysis on a napkin would tell you you need to screen 400 animals per group to detect a 5% drug effect in this commercially available model from the JACs. I said that recently at an ALS meeting because 55 labs have now ordered mice from the JACs, and there was a hush in the audience, leading me to believe people are screening drugs in this model, and you can't. The variability in survival is way too big, unless you're going to do 400 animals per group, which nobody's going to do. So what is it going to take to get there? How are we going to take another decade to characterize this model like we did with SOD1? And is it really even a model of neurodegeneration? How reproducible is it? Are the molecular mechanisms similar to SOD1? That's taken over a decade to figure that out. We still don't really know what the gain of function is at this point. But we have to figure that out because this could be a really crucial model for the field for drug development with, as somebody said earlier, a significant unmet need. So it's not a good screening model. There's no way that this is a good screening model because of the variability. It's 150-day variance in survival. So that has to be one of the goals when you start pushing this model forward for drug screening, is how are you going to improve the model so it doesn't have as much noise. And this is the recent data from the publicly available colony that's at JACs. And you can see how different the kinetics are between males and females. So this colony, as is commercially available, is not a useful drug screening paradigm for us. So we put together a proposal to try to shorten that time frame of 10 years and to really do this through collaboration. Nobody could do this alone. So we went out there and said, look, we'll take an ownership of it and we'll put together a plan to characterize that model and answer some of those questions and try to improve the reproducibility of the model. So in order to do the whole thing, we estimated it was going to be about $1 million, which was quite a lofty goal. First thing was to generate a congenic. It was only a 4x back cross on the black 6, and that's probably why it was so noisy. To do a power analysis, we had 6,000 animals in that slide with all of the red dots for SOD1 that allowed us to identify those four variables. We're estimating we needed about 1,000 animals that had been born at our facility so that we could start to understand the kinetics of the disease in that model. We wanted to look and create a phenotypic scoring system. If you have to sacrifice the animals to see if a drug is having an effect or not, that would be quite challenging. So we wanted to see if it had progressive neurodegeneration like we see in the SOD1 model. We have a scoring system for that model because we've been looking at it, and we've probably seen 8,000 or 9,000 animals pass through our facility in the last 10 years. But can you observe this animal model in some fashion and show a progressive phenotype which hadn't been described in the publication? And finally, ultimately, since we want this for target discovery and drug screening, we wanted to profile a bunch of tissues in this animal model, much like we've done with a couple of other models of neurodegeneration. And those experiments in our hands are quite complex, and I'll just describe what we've done for the SOD1 model and another model of neurodegeneration called LOA. The SOD1 model, the mice look totally normal until about day 70 when you start to see paralysis in the tail. From there, it's progressive. About 15 to 20 days later, you'll see paralysis in the hind limbs. About 15 days after that, you'll see paralysis in the forelimbs. And again, most of the animals can't move around the cage, and we euthanize around day 135 when they can't right themselves. We started molecularly profiling that animal at day 30, and at 10-day intervals all the way to day 135. And we didn't just harvest spinal cord to do gene expression profiling and proteomics. We actually harvested a dozen tissues out of that mouse. And out of that has come some really interesting drug discovery projects, which I'll show you a little bit about. So this profiling experiment is the vast part of this proposal. It's really characterizing phenotype and looking at molecular mechanisms. So we basically got together a group of stakeholders, MDA, ADDF, and AFTD. Obviously these because TDP43 mutations affect that patient population, and basically said, can we get there? And we're about 12 months into the project at this point. And we've done a couple of really good things. Here's the kinetics of our colony at TDI now that it's a 12x transgenic backcross onto black 6. The kinetics are much, much better than what I showed you previously. And within a gender, you certainly can, it's actually tighter than SOD1. It's only about a 7-day variable in survival. So you can actually now do drug studies in this animal model with about 22 animals per group and detect 5% drug effects, which is outstanding. We are out to about 1,000 animals now, so we'll be able to do that power analysis and actually publish a formal study design. If you want to screen drugs in this model and detect that drug effect, here's how you have to power it. Things like monitoring the transgene, all of those things will be characterized as part of this study. We have developed a phenotypic scoring system. These animals are not as progressive as what we see in SOD1, and I'll tell you about that in a second, but you can see changes in polyclastin that are very reproducible. When you pick them up, they do trunk curling that's very reproducible. So you actually don't have to euthanize these animals to start to see drug effects. You can actually phenotypically score them. Again, they're much more subtle than SOD1. In fact, the first few animals that we had at the colony that reached end stage, they looked totally normal, and then 2 or 3 days later, all of a sudden, they succumbed to the disease, and I'll show you why in a second. So what's going on, though? Is this a model of neurodegeneration, which is a good question? Well, one of the first things we look at is the hallmarks of astrocytosis activation. Here's wild-type control animals stained with GFAP, a marker of astrocytes. Here's SOD1, and here's the TDP43. So there is some astrocytosis. It's not as prominent as what we see in SOD1, but it is there, and it's there about 25 days before they succumb to the disease. Out at the neuromuscular junction, for folks that have never looked at staining of the neuromuscular junction, you can stain the neuron coming into the muscle beds with one dye, which here is colored in yellow. You can label the neuromuscular junction, which is where the neuron attaches in the muscle, with bungarotoxin. And what's shown here is SOD1 at day 100, and you'll see 3 types of NMJs here. This is a fully occupied NMJ, which is normal. Here's an NMJ that the neuron has died back and it's not overlaying the pretzel shape. And here's one that's completely died back. So in TDP43 at day 80, and again, this is about 35 days before end-stage disease, there's a lot of normal-looking NMJs, but there's also NMJs that look like this, which looks just like some of the ones we see in SOD1. So there is denervation out in the periphery in this model, just like we see in SOD1. Finally, we have developed some IPS lines from this model, which are actually quite interesting, and that's why I'll throw them up here. At 37 degrees, the mutant human protein, which is labeled in red, is completely nuclear, just like where it should be when it's non-mutated. However, when you heat shock these guys and move them to 42 degrees, all of that mutant TDP43, the human transgene, all goes out to the cytoplasm, which is what's been described in many of the other models. If you switch them back to normal temperature, all the mutant human TDP43 goes back and becomes nuclear, and the mouse genes never translocates to the cytoplasm, at least so far with all the assays that we've tested, including hydrogen peroxide and a bunch of others. So it could be potentially an interesting model to think about for high-throughput screening purposes. These animals don't die of neurodegeneration, at least not yet. They die of a bowel obstruction, and that's why it's so acute. As you can see, they're just literally filled with poop. Which we were very, very surprised about. So one of the things that we're trying to do is get rid of this issue and see if we can get the animals to live longer by getting their bowels moving again, and maybe then they'll succumb to the disease by progressive neurodegeneration, but at this time, that's really not what they're succumbing to. So what does the future hold, at least for us? We're really committed to animal work, obviously, as you can hear from my presentation. The other thing that was a theme from this morning, which I thought was fantastic, and I'll emphasize it, is we do all of our pharmacokinetics in SOD1 mice. Even though that's a lot more expensive because it's a transgenic animal, we have seen significant differences in PK and biodistribution for protein biologics and small molecules in this model compared to C57 black 6 strains, for instance, and that's not surprising. These animals, depending upon what drug class you're talking about, have full-blown immune system activation. So those would create sinks for drugs that are targeting that system, and so therefore your PK and biodistribution changes quite a bit. So we recommend for everybody that's working in models like these to do all of that upfront work that's been talked about this morning in your model of interest before moving into survival and efficacy studies. The other thing that I didn't include in my deck, but we have an incredibly large database at this point, probably 75 drugs. We do full PD studies in parallel to all of our survival efficacy studies, where we bring animals down at day 85 and 100 that have seen drug as well as controls for full Affymetrix profiling, proteomics assays by Luminex as well as mass spec, histology like I showed you for axon counts, neuromuscular junctions, and so it's quite a large database of drug effects in the model and what those drugs are doing from a systems biology perspective. So we need more models though. The SOD1 model is the only model in my mind that has been validated in this field. And I would challenge anybody to say that a PD model has been validated at that level, an AD model has not been validated at that level, an MS model has not been validated at that level. And if you look at the publications in the field for a lot of those animal models, depending upon the investigator, the kinetics of survival are completely different from one lab to another. We had one group that asked us to come visit their facility because every single drug that they were testing seemed to increase survival, and their median survival of their colony untreated was 110 days, and every single drug they screened it was 135. Well, we started doing blood chemistries on their animals, and every single animal was so contaminated with bacterial infection, they were just basically curing that with a lot of their immune modulatory drugs that they were testing. So those are the types of things that I think you have to look at. Obviously, models need to be validated, but they need to go through this process of rigorous validation before you can roll them into a drug screening system and protocol, or else your false positive rates are just going to be exorbitant. And I will close there and open it up for questions.
Excellent talk, and I applaud the foundation for delving into this too deeply. You showed a couple of examples of false positives. Jilly Burgess from Pfizer did the same thing in pain, where they took a couple molecules and found out it was the methodology. What I really worry about are false negatives. I was hoping if you could comment on that. And the second thing is, what's your view on multifactorial models? In other words, in the human condition, you have a human mutation, but the expression of that may depend on other risk factors, et cetera. So in Parkinson's disease, why not take a synuclein model and do a GBA on top of it, because it's a known risk? What do you think about double mutants and what problems that have?
So two different questions. I don't think, for instance, that we worry about false negatives as well. We think we do an outstanding job at this point of false positives. False negatives are a little bit more challenging, and I agree with some of the sentiments of folks saying, if you really believe on your mechanism, we don't think you have to make a go decision, no-go into the clinic based on improving survival. It's a nice have. But actually tomorrow at the BioCEO Investor Conference, I'm going to present that we're going to push a drug into phase 2 this year that we're repurposing from another disease indication. And at this point, it does not statistically change survival in our hands in our mouse model. It trends towards improving body weight. It trends towards slowing down disease progression. It trends towards survival, but it doesn't statistically affect any of those things all the way. And we've only done one dose. My argument is if we played with dosing in the animal model, we'd probably get there. But we know from biomarker analysis that it's doing a lot of exciting things. It's knocking down neuroinflammation in the CNS, it's knocking down neuroinflammation in the periphery, which right now is a biomarker for a phase 2 clinical trial in ALS. It's improving NMJ occupancy. So it's doing lots of good things at the biomarker level. So we're going to push that forward into phase 2 without improving survival. So I think the survival is a nice have, but I don't think it's a requirement. As far as pushing multiple either genetic models together, we're doing that. We're actually crossing the LOA mouse into the SOD mouse. Interestingly enough, there is one publication that has done that, and instead of exacerbating disease, it actually is the biggest effect I've ever seen in an SOD1 model as far as improving survival. So I think there's some value there, just like there's also value in cocktail approaches when you test preclinically, but as my head of pharmacology loses his hair, is doing those types of experiments in animal models is really, really challenging to try to predict the right doses for additive or synergistic effects. It's not always a one-to-one relationship. So it's a harder road forward. We just have to be careful.
Yeah, great work. One of the things that's come up in discussion is really the value of behavior, especially in neurodegenerative disease. In the case of ALS, I mean, you eventually found that, you know, survival wasn't related really to behavior at all, I guess. But even if you solved that, I wondered what your views were. I mean, in the Alzheimer field, you know, mice don't write books, so we always have that kind of comment on the behavior. But you know, these are mostly target-based models. In other words, we want to see if the drug is hitting the target on all of the various biomarkers that you hit. And I just wonder, since behavior is so expensive and variable, whether you think it's really important actually in making go-no-go decisions.
It's really important. There's no doubt about it that it's critical. And I think, again, one of the things that you saw in some of the work we've done, which we've tried to replicate, is we actually have tried to get closer to what is really judged upon in a clinical trial and push that back into our animal work. Everything that we do has really, really large N's. Like when we count NMJs in a drug study, we don't do that with 2 mice. We do it with 18 mice per group. And we don't just count a field of NMJs. We count every single NMJ that's in the calf muscle. When we count roots from all 18 animals per group. So I think those are the things that you have to do if you're going to convince yourself that the effect that you're seeing in your preclinical animal model is real. Now, what hope that has to translating into the clinic, especially in a disease like ALS when there isn't anything there yet, we don't even know what that road looks like because it's never been paved, it's hard to predict.
I mean, I guess what I'm asking is if you're counting every neuromuscular junction, you know, why does it really matter if they're slowing down in the cage or not? You know, how much more value do you get out of that?
Well, I think the value there in my mind is, out of everything that I presented, the most predictive biomarkers that might translate into the clinic and be meaningful are the ones that we think improve function, right? So let me give you an example of that. We've treated SOD mice with a drug that increases body mass and body weight 4x, right? So that's one of the hallmarks of ALS and the disease is muscle atrophy and muscle wasting. It's progressive, right, starting at symptom onset. Those mice don't live a day longer and it doesn't slow down onset of progression by a day. So you want to look at what you think are the functional outcomes. And in a disease like ALS, it's where the rubber meets the road. If you have completely denervated muscle, the muscle's not going to work. And that drug that actually improved muscle mass, the denervation was exactly the same as an untreated animal. So we think axon counts, which obviously you can't do in people, and we think NMJ occupancy, which we're starting to get in that direction. I think that might be able to translate some of those types of things to the clinic, measuring them a little bit differently. Those are the things that I think are important to predict whether a drug might translate or not.
Excellent talk, Steve. You've obviously done some very beautiful work. One of my questions is ALS, how many forms of ALS are there? And when I say that, you talk about SOD1 mutation. Did all the human patients that didn't respond have similar SOD1 mutations? 2, did they show the same sort of similarities? Because I think of ALS and many of the neurodegenerative diseases very heterogeneous. I'm a veterinarian, I work with dogs, and I can tell you there's much difference in the presentation of spinal cord degeneration that we see in dogs among the different breeds. And so we're thinking that although they all manifest the same way, the back end stops working and the gut gets flaccid, as you pointed out with your TDP-43s. It's very interesting. Anyway, beautiful work. Sorry.
A lot of questions there. Let me try to answer a couple of them.
I have a lot of questions.
It's a really heterogeneous patient population. The clinical presentation is actually pretty similar, but that's just because we don't have biomarkers for diagnosis or even to subset patient populations at this point. So that's a problem right there in and of itself. Is the genetics, it tells us a little bit. Only about 10% of ALS cases are genetic in nature. Because of next-gen sequencing technologies, we are now breaking that barrier down really, really quickly.
In humans, is that all single copy in
yes,
the natural patient?
for the most part. And so from the genetic standpoint, we'll probably have all the genetic mutations identified here in the next couple of years because it seems like we're at the pace of about 2 or 3 new ones per year now. That's still going to leave a vast majority of patients that are sporadic in nature. It's not found in their families, and we don't know what the underlying genetic cause is going to be. Maybe if we sequence enough of those sporadic patients, we'll start to get some clues. Certainly RNA processing and editing at this point is becoming a common theme. We don't really know what that means, but we know that TDP-43 binds to 6,000 different RNAs. in the cell. We know that FUS1, another common mutation in ALS, about 2% of cases, is also an RNA binding protein. And we know that a new mutation in an ORF also appears that it might be an RNA sink and that has a trinucleotide repeat and an intron that could be messing with normal RNA processing and leading to problems with probably protein production and protein folding ultimately.
Anyway, we'll talk later. Excellent
Sure.
presentation.
So I wondered if you could sort of comment upon, the fact is you as a foundation have been able to put the money up front to develop this so that the community can benefit from using a model that can then potentially give them results that they can rely upon and go further in the process. I wonder if you could sort of comment about how you could make perhaps a financial argument to say that if a new model is made, people should actually refrain from using it until this sort of rigor has been applied to it, so that it actually makes sense from a funding perspective to ensure that models are developed with this level of rigor, akin to also what the pamphlet we received outside from the ADDF. Can that be made? I mean, could that argument be made?
I think it has to be made. We don't really believe any drug studies we see in animal models unless they're really, really well done because of the noise in these biological systems. So I think it has to be done. I think the challenge is finding out who's going to pay for them, because it's not trivial to take that task and to do it. It's not really sexy work, right? It's grunt work for the most part, and most organizations and academic labs don't have the finances and bandwidth to do what we've done. And we're driven by a selfish motivation. As a nonprofit, our mission is to get drugs into patients, and unless we have good tools, we're not going to accomplish our mission, so we're willing to invest in the space. But making this a standard practice, I think, will be a little bit challenging from a financial perspective.
But the argument is that if you can show that doing it this way actually saves money in the long run, and you can make that argument,
you can.
Then you have to say, don't fund all of these studies that are coming to, let's say, funding bodies that are doing it in N's of 4 and this, that, and the other. You just send them back and say, get the model sorted out first, and then we'll fund these studies.
I think data like this is a driving force for that decision.
I enjoyed your presentation, and I had just one quick question about the observation of the mice, euchogenic line dying from bowel obstruction, and I was wondering if you looked into the underlying basis for that. Is that due to, for instance, the innervation of smooth muscle cells and lack of peristalsis, or is that consistent with some of the neuromuscular effect possibly, or totally unrelated?
So we haven't gotten that far yet. So I don't have an answer to your question. At this point, we're actually focusing more on the brain biology now. Obviously, this is a mutation that affects not only ALS patients but also FTD patients. So we kind of jumped from doing some of our spinal cord muscle stuff, which we standardly do for SOD1, and we started to look and see what's going on in the brain of the mouse, particularly the motor cortex, and see what's happening there. We are trying to deal with that bowel obstruction piece, but it's kind of secondary for us, and it's out of our sweet spot. I mean, our goal here is to understand if this is a good model, A, for drug screening, which I think we can give it a checkbox now because it's pretty reproducible, but B, is it really a model of neurodegeneration that's useful? And that one, I'm still putting a question mark there. I don't think we're quite there yet. So I don't know the answer to your question yet. Thank you.
I'd like to thank the ADDF for the invitation to speak today, and that last talk was fantastic. And I want to indicate that I've been advocating similar things in the AD field with far less traction and backing than we just saw for ALS. I'm going to take it to the next level in this talk, but please keep the context of the last talk in mind as I step through this. Translational research, what is it exactly? We have this vectorial paradigm where we've moved from preclinical research to clinical research with one informing the other, with the preclinical being target identification and validation, whatever validation means, lead identification, optimization, pharmacokinetics, pharmacodynamics, ADMET, and in a disease model, and that informs the clinical trials. And that's where we get into fluid and imaging biomarkers. I'm going to be emphasizing points about neural network functioning, not just single targets. But the reality of this is that should be a two-headed vector, because we should be taking what we learned from the clinical research and reapplying it to the preclinical realm in order to refine the animal models and the applications, as well as networks and discoveries, discovery science. And this is an overriding theme that you'll see through this talk. So, issue number one, PK/PD and ADMET. Few pedagogical statements. In vitro experiments are static with respect to target and the compound under investigation. In vivo experiments are dynamic with time-dependent impacts on compound concentration, distribution, and target disposition. You can't just take an in vitro active compound, regardless of its potency. We have picomolar inhibitors, by the way, I can brag about that, and say, I'm going to test this in animals. Because how will you know if your target is reached at an appropriate concentration and if the pharmacodynamic readout is due to the impact of the compound on the target? How long will there be a pharmacodynamic effect if you get a delivery of a compound at whatever level to its target? So selected aspects of PK, PD, and ADMET must be considered in the strategic design of in vivo experiments, and biomarkers are required for performing these pharmacodynamic analyses. Keep in mind, all human clinical studies are in vivo experiments. So this is the standard background on PK, PD, and graciously I don't have to go through any of that except to point out, which is my laser. Here's, uh-oh, I hit the wrong button. Yeah, okay, cool. So you have your PK where you have a compound that's going into the system and it's diminishing, it's being metabolized, it's being excreted, it's being removed from the system, and you have a pharmacodynamic effect which peaks at a certain time and then diminishes as the concentration of the drug in the in vivo system is diminishing. So PK relationship is the relationship between the dose of a drug and changing concentrations of the drug in the body of the tissue. Pharmacodynamics is the relationship between the drug concentration at the site of action and the effect that is elicited. And the classic statement, which came from Louis Scheiner in 1981, if I can do my arithmetic, that's 31 years ago. Yeah. Pharmacokinetics is what the body does to the drug. Pharmacodynamics is what the drug does to the body. ADMET, we've heard before, absorption, distribution, metabolism.